Okay, so here we're going to talk about the digestive system, some of the functions of the digestive system. You've got ingestion, so that's just actually physically bringing food into the mouth. We're going to start the very early stages of digestion in the mouth. Motility, you've got both voluntary and involuntary muscular contractions. We're going to use those kind of for two different things. One is for mixing, because you got to mix a bunch of the enzymes and the acids along with the food. In addition to the physical movement of, moving the food from the mouth down through the GI tract towards the anus. Secretion, which is the process of releasing different products. So there's different enzymatic products that we're going to have to release, things like bile. We'll get to all that, but secretion is definitely part of the process of digestion. And then actual digestion, which is just the breaking down of the food into smaller bits and pieces so that we can actually absorb it. We do that both mechanically, and if you want to think of like chewing as mechanical digestion, and then physically where we're using enzymes to make it smaller. Absorption is the movement of these smaller nutrient molecules from the inside of the GI tract into our blood so then it can be distributed. And then elimination is the expulsion of all of the components that we took in that we didn't digest and absorb. So the organization of the digestive system, you've got the GI tract, and then you've got some accessory organs and accessory glands. So I don't want you to confuse the terminology between digestive system and GI tract. When you're talking about the GI tract, you're talking specifically about the tube or the actual pathway that the food is going to take. So you're basically going from the oral cavity to the pharynx or the throat, the esophagus. I got to jump behind the liver here, the stomach, the small intestines, the large intestine, and then out through the anus. That's the GI tract. That's the tube or the pathway that the food's going to take. Now, along the way, you're going to get contributions from different accessory glands and accessory organs. So your salivary glands, your liver, and your pancreas, these are more of accessory glands, and I'm using the term glands because they all play a role in releasing something, whether it's bile or pancreatic juice or saliva, that helps in the process of digestion. Then you've got other structures that aren't necessarily accessory glands, your teeth and your tongue, your gallbladder, things like that, have to do with, assisting in digestion, but they're not part of the GI tract. The gallbladder is iffy because it's not really considered a gland because it's not releasing something, but it kind of does. It's a little tricky because your liver produces bile, and then it goes into the gallbladder, and then the gallbladder kind of contracts to make it come back out, if that makes sense. But it's not being originated, the bile is not being originated in the gallbladder. It's more of a storage tank, and it concentrates the bile. Okay, so the GI tract, all the way from the esophagus through the large intestine, is a hollow tube, and it's made up of 4 different layers. So I will try to kind of point these out in some of the different images that will come up in this PowerPoint. But the mucosa is the first layer. It's got 3 different parts: the epithelium, the lamina propria, and the muscularis mucosa. I'm not going to point out these smaller subcomponents on this slide because it's not the best one. I'll try to do it one of the later slides when we've got a bigger picture. Submucosa, you've got the submucosal nerve plexus that innervates that. Muscularis layer is a muscular layer. It's got an inner circular layer, so this is the inner layer. You can see the fibers of the muscles are kind of oriented circumferentially or going around the lumen. And then you've got your outer longitudinal layer. This is the outer layer, and you can see these muscle fibers are running more lengthwise. And then your last layer is your serosa. Some tissues have an adventitia, is basically the same thing as, there's a subtle difference between adventitia or serosa. I'm not going to get hung up on the details of those. It's essentially just that outer layer that wraps all of the other layers kind of together. Okay, so let's look at the mucosal layer. It's composed of 3 different parts. So you've got your epithelial layer, which is a simple columnar epithelium that makes contact with the lumen of the GI tract. Your lamina propria, connective tissue layer. Right underneath that epithelial layer, you've got blood vessels and you've got nerves in there. Once we break down our food to its smallest component, those nutrients are going to get absorbed. They're going to cross over the epithelial layer and they're going to go into the blood vessels that are laying in that lamina propria. Then the third one is your muscularis mucosa. This is a very thin layer of muscle under the lamina propria. It's really thin. It just does these really kind of slight contractions, and it helps to facilitate the release of some of the mucosal secretions into the lumen. So we've kind of got some different things that get released into the open space of the GI tract to help with digestion. And contraction of the muscularis mucosa helps to kind of squeeze those contractions out, or the enzymes and secretory products out. Okay, after the mucosal layer, you've got the submucosal layer. You've got larger blood vessels, lymphatic vessels, and nerves in there. So here is, let me point out the mucosal layer here. You've got this is layer 1, your epithelial layer. This is layer 2, the lamina propria. This is layer 3, the muscularis mucosa. You can't really see the red on red. And then this layer right here is going to be your submucosa. Nerves that innervate the smooth muscle and glands of the mucosa and submucosa make up something called the submucosal nerve plexus. Connective tissue in both the lamina propria of the mucosa and the submucosa have that mucosal associated lymphoid tissue there. In the last region of the small intestine, which we'll get there, but that's called the ileum, you've got larger aggregates. That's where you find your Peyer's patches that we've talked about. And again, that mucosal associated lymphoid tissue is just kind of lymphatic tissue, kind of packaged with a lot of immune cells to help catch any microbes that may have been taken in through the diet, that maybe survive the acidic environment of the stomach, and we want to kind of catch those in that lymphatic tissue before it gets into our bloodstream. The next layer is the muscularis layer. So this is much thicker than the muscularis mucosa that was part of the mucosal layer. The muscularis layer is mainly there to do 2 main functions. For one, it's going to mix the contents, so you get this kind of bolus of food. It becomes chyme once it's mixed with the contents of the stomach. And then it goes into the intestines. And in the intestines, we need to be continually mixing that with digestive enzymes so that we can continually digest it. So the contractions of this layer will help to mix, but it also is involved in propulsion. So it goes from the stomach into the first part of the small intestine, and we've got to basically make that travel through the remainder of the small intestine and then the entirety of the large intestine. So we have these what are called peristaltic contractions that essentially push that segment of chyme throughout the GI tract. So these muscles are supplied by a group of nerves known as the myenteric plexus. The submucosal nerve plexus, together with the myenteric plexus, make up what's called the enteric nervous system. That is basically the nervous system of the GI tract. So the muscular layer has two layers of muscle. The inner circular layer, I pointed that out earlier, is oriented circumferentially around the GI tract, and I'll kind of show you this again in a minute. And then the outer longitudinal layer is oriented a different way, meaning that the muscle fibers are running in different directions, and that really helps with the mixing. The inner circular layer is thickened at different regions along the GI tract to form a sphincter, and a sphincter essentially closes off the lumen between two points in the GI tract, and then it can open to release contents from point A to point B. I'll point one of those out when we get to the end of the terminal region of the stomach. Before it gets to the small intestine, we have the pyloric sphincter. That's an example of one. And essentially what the sphincters do is they regulate the passage of the material from one point to the next, so it doesn't all just flow through rapidly. Okay, these are those two types of contractions that I was talking about. The actual picture that's shown here is showing you peristalsis. Well, I don't have a picture of mixing on this slide, but you can see that you've got these contractions that's pushing the bolus of food or the chyme. It's starting at the top, it's moving down, and that's essentially how we move the material from point A all the way towards the anus. Mixing is the back and forth or kneading motion that happens, and again, the point of that is just to mix the chyme with the different digestive enzymes so we can get maximum digestion. Serous membranes of the abdominal cavity. So in the last lecture we talked about serous membranes of the lungs. That was called the pleura. In the abdominal cavity it's called peritoneum. So like always, you've got the visceral and the parietal. Parietal peritoneum is the serous membrane that lines the inside surface of the body wall. Visceral peritoneum is a serous membrane that covers the actual surface of the organ. And then between the two layers is your peritoneal cavity, and you've got that serous fluid there, that lubricating fluid that exists in the cavity. For the abdominal cavity, we've got a bunch of different organs, as you know, that are in there, and so we kind of classify them as intraperitoneal versus retroperitoneal. And the difference is how much visceral peritoneum do they have on their surface. So intraperitoneals are organs in the abdomen that are completely surrounded by the visceral peritoneum. That's going to be the stomach, the majority of the small intestine, and then parts of the large intestine. Now, retroperitoneal organs kind of lie more against the posterior abdominal wall, so only the front and the sides of them are covered with visceral peritoneum. So that includes a majority of the duodenum. The duodenum is the first part of the small intestine, the pancreas, both the ascending and descending parts of the colon, and then the rectum. So all of those lie more towards the posterior wall of the abdominal surface, so the visceral peritoneum only covers the anterolateral, or the front and the sides of it. So the back portion is bare of that visceral peritoneum. There's two main types of receptors you're going to find in the GI tract. Mechanoreceptors detect stretch or pressure. That's going to be important for knowing when these organs are full of content. And then chemoreceptors detect specific chemicals in the GI tract. Activation of either of these receptors is going to send information to the central nervous system, and then the neurons will respond by activating different glands and pancreatic secretions, in addition to stimulating muscle contractions. So essentially it lets the brain know that something's in there, and then the brain sends messages down to the muscles and the glands to say, okay, do what you got to do to get digestion going. The upper GI tract is where initial processing by both mechanical and chemical digestion is going to take place. It includes the oral cavity, And salivary glands, I'm highlighting this. This is misleading because this is not part of the actual tract. Like I said earlier, it's an accessory gland that's associated with the GI tract. Does that make sense? So the oral cavity, the pharynx, the esophagus, and the stomach. That's all part of the upper GI tract. So we're going to go into a little more detail about the components of the upper GI tract. You've got the oral cavity, which includes the salivary glands. So the oral cavity obviously is the mouth. It's going to receive secretions from the different glands continually, so not just when we're eating. Obviously, it's going to ramp up when we're eating because it's got a bigger job to do, but we are receiving continual secretions from the glands, all the time. So we've got a bunch of unicellular glands associated with the oral cavity, and those are collectively called our intrinsic salivary glands. Secretions of the intrinsic salivary glands, one of them is called lingual lipase. So we are going to remember this one. So lingual has to do with the tongue. And I just realized I don't know how to spell tongue. I don't know where the U goes. So it's somewhere in there, there's a U and an E. Okay. And then lipase. Anytime you see the word lipase, you think of digesting fat. Because there's going to be a couple of different lipases. So when you hear lipase, you know that digests fat. And if it's lingual lipase, then it's associated with the tongue. So you can then tell me that lingual lipase is the enzyme that comes from the oral cavity that digests fat. I'm telling you that now because we're going to talk about a handful of different enzymes. And if you understand the terminology of their name, it will help you remember where they're from and where they act. Okay, so lingual lipase was part of the intrinsic salivary gland secretion. The majority of the saliva, however, is produced from these larger multicellular exocrine glands outside of the oral cavity called our extrinsic salivary glands. For the extrinsic salivary glands, we've got the parotid. We've got the submandibular salivary glands, and then sublingual under the tongue. The parotid salivary glands are going to be the largest ones. Each parotid gland is located anterior and inferior to the ear, so you can see it here. Infection of the parotid glands by a specific virus can cause mumps. Children are vaccinated against this when they get their MMR vaccine. Submandibular glands here. Both inferior to the floor of the oral cavity and medial to the body of the mandible, so submandibular, that's kind of where their name comes from. The submandibular salivary glands produce the majority of the saliva. I would probably remember that. And then you've got your sublingual salivary glands. They are, as the name suggests, underneath the tongue. Each one, they've got ducts that open to the inferior surface of the oral cavity. Okay, the volume of saliva secreted daily can be anywhere from 1 to 1.5 liters. Most of the saliva is going to be produced during mealtime, but of course there's smaller amounts that are produced continuously. Saliva is mostly water, 99.5% water, but in addition to that, it's got salivary amylase. So this is another one of those enzymes. Salivary tells you where it comes from. Amylase means the breakdown of starch. So just like lipase had to do with the breakdown of fats, amylase has to do with the breakdown of starch. So salivary amylase is the enzyme that breaks down starch that starts in the oral cavity. It's also got mucin. I think I mentioned before that mucin, it combines with water to form mucus. And then lysozyme, which is that kind of natural antibiotic that we have. So the combination of all of those together allows the saliva to do a couple of different things. So for one, it moistens the ingested food to help it become a bolus. A bolus, as gross as this sounds, is a globular wet mass of partially digested material. That makes it easier for us to swallow it. Initiates the chemical breakdown of starch in the oral cavity because of that salivary amylase. So there's not a whole heck of a lot of digestion that goes on in the oral cavity, but we do start the breakdown of starch there because we've got the salivary amylase. It acts as a watery medium into which food molecules can be dissolved so that we can stimulate our taste receptors. Cleanses the oral cavity structure, and then it helps inhibit bacterial growth because it's got that lysozyme and there's certain antibodies in there as well. Mechanical digestion, this is chewing, or at least in the mouth is referred to as chewing, otherwise known as mastication, serves to mechanically reduce the bulk into smaller pieces. So basically we can't just put a huge chunk of food in there and swallow it. We've got to make it smaller and then create it into that bolus. So this is going to require activities of your teeth, the skeletal muscles, and your lips, your tongues, your cheeks, and your jaws. We've got a certain region in the brain called the mastication center that help to coordinate all of that activity. A little bit about the anatomy of the tooth. You've got the crown. This is the exposed region. It kind of comes inwards right here. This is known as the neck. Dentin forms the primary mass of a tooth. So all of this is composed of dentin. Dentin is very similar to bone, but it's actually a little bit harder. And then the external surface of dentin is a durable layer of enamel that forms the crown of the tooth. Enamel is the hardest substance in the body. It's mostly calcium phosphate crystals, so again, similar to bone, but it's actually harder. So the crown is what's going to be made of enamel. And then in the center region of the tooth is the pulp cavity that contains a connective tissue called pulp. So this would be your pulp cavity and all of your pulp in there. Okay, we get 2 sets of teeth during our lifetime. In an infant, you get 20 what are called deciduous teeth that are up anywhere between 6 months and 30 months after birth. As you well know, those teeth are eventually lost and replaced by 32 permanent teeth. So this one shows you, this is a child's mouth, this is an adult's mouth. The most anteriorly placed permanent teeth are called incisors. We've got 2 central incisors and then 2 lateral incisors. Immediately posterolateral to the incisors are the canines, which are also called cuspids. So there's a canine, and there's a canine. And then you start your premolars. So you've got your first and second premolar, and then you've got 1, 2, 3 molars. The molars are going to be the thickest and the most posteriorly placed teeth. You can see the anatomy of the molar here. They've got a large, broad, flat crown with distinctive cusps, and then they have 3 or more roots right here, rooting into the gums. The pharynx and the esophagus, so the pharynx and the esophagus eventually will connect the oral cavity with the stomach. The pharynx is the funnel-shaped pathway for both food and air. It's made up of skeletal muscles that form the wall of the pharynx. And then it opens into the esophagus. The esophagus is a muscular tubular structure that is normally closed, but will open if we're passing food through there. So pharynx here, and then esophagus. And the esophagus is going to penetrate through the diaphragm. You can see the diaphragm is here. The esophagus is going to penetrate through and then open up into the stomach. There's a ring of circular muscle at the superior end of the esophagus. It's called the superior esophageal sphincter here, which closes during the inhalation of air so that air doesn't go down the food pathway. And then we also have an inferior esophageal sphincter here that closes off, and it opens when the food empties from the esophagus into the stomach, but it's there mostly to prevent the acidic acids of the stomach, acidic contents of the stomach rather, from entering the esophagus. So I'm just going to try to summarize this clinical view. It's about reflux esophagitis and gastroesophageal reflux disease. So I talked about that lower esophageal sphincter, that muscular ring that closes off between the lower end of the esophagus and the upper end of the stomach. The stomach is very acidic. It needs to be that way so that we can kill the bacteria that come in through our food, so that we can digest the food and break down proteins in the food. So it kind of has to be really acidic. And the stomach is basically equipped to handle that. So we'll actually look at that here in a little bit and see some of the mechanisms that the stomach uses to protect itself. The esophagus, however, is not equipped to handle the acidity of the stomach. And so ideally, the contents of the stomach should not come into the esophagus. But things don't always work that way, and sometimes you do get the reflux of some of the acidic stomach content into the esophagus. So that actually presents as kind of a burning sensation, and because of the location of the esophagus, it presents in what people perceive as to be the heart. That's why we call it heartburn. It's actually the esophagus, but because it's close in proximity to where the heart lies, it's felt in that region. So some of the risk factors for this are being overweight, smoking, if you eat an extra large meal, larger than you normally would, hiatal hernias, where there's a little portion of the stomach that kind of protrudes up through the diaphragm, which normally it should not. Spicy foods, too much caffeine, things like that can all kind of increase the risk of reflux. Chronic reflux esophagitis, so if this is happening over and over, can lead to GERD, which is gastroesophageal reflux disease. And basically, you're getting so much of the acidic contents that you start to damage and erode the lining of the esophagus, and it can lead to scar tissue. If that continues and you get to more advanced stages of that, then you get something called Barrett esophagus, where you've sort of eroded the lining of the esophagus, and now you start to change the epithelial type. So the normal esophageal epithelium goes from stratified squamous to columnar secretory. And that's what we call a metaplasia, where you change your epithelial lining. And so that puts you at a increased risk for cancer. So some of the ways that we can help this, we can use proton pump inhibitors, Prilosec, Nexium, and those work to reduce the acidity of the stomach a bit. There's direct antacids like Rolaids to help kind of neutralize the stomach acid. So basically the medications are working to reduce the acidity of the stomach contents so that if it does come up, it causes less damage. This is why they tell you don't lay down directly after a meal, because gravity does its part to keep the stomach contents in the stomach. If you eat a meal and then your stomach is activated, so we're releasing a lot of that acidic contents, and then you lay down, it's just going to be an increased chance that it's going to come up into the esophagus. Swallowing is called deglutition. It's a process of moving the ingested materials from the oral cavity to the stomach. There's 3 phases. The voluntary phase, the pharyngeal phase, and the esophageal phase. Voluntary phase is happening in the mouth. So voluntary phase is happening in the mouth. We are consciously controlling this movement as we're chewing, and we're pushing that bolus of food back towards the throat. Pharyngeal phase is involuntary, so once we swallow, it's kind of out of our hands in terms of conscious perception of what we're doing. Pharyngeal, so we're in the pharynx. Here's your bolus of food in the pharynx. And then the esophageal phase as it travels down the esophagus, also involuntary. Okay, so our bolus of food has finally made it into the stomach. So the stomach is a holding sac. It's in the upper left quadrant of the abdomen. You can see it here. Immediately inferior to the diaphragm. Both mechanical and chemical digestion will continue to happen in the stomach. Specifically, with respect to chemical digestion, we start the digestion of protein and fat in the stomach. So the vast majority of our digestion and absorption is going to continue to happen in the small intestine. But you see that we got a little bit of starch digestion happening in the oral cavity with that salivary amylase. And we're going to start to get digestion of protein and fat in the stomach. So ingested nutrients will spend between 2 and 6 hours in the stomach. Fatty substances tend to take longer to be emptied. Absorption in the stomach is minimal. Small nonpolar substances can get absorbed, but the vast majority of absorption takes place in the small intestine. The internal stomach lining has many gastric folds. You can see those here. All of these wrinkles, these are called rugae or gastric folds, and they allow our stomach to expand as we take in food and drink and then return to its normal size. So gross anatomy of the stomach, you've got the cardia. The cardia is this small kind of narrow entryway from the esophagus into the stomach. The fundus is this dome-shaped region here that projects kind of superior to the esophageal opening. It kind of makes contact with the surface of the diaphragm. The body makes up the majority of the stomach. It's going to extend from the cardia all the way down to the pylorus. And then the pylorus is this area here, this narrow funnel-shaped pouch, forms the terminal region of the stomach. It opens into the duodenum. Here's the duodenum, which is the first part of the small intestine. So this right here is small intestine. And then you've got your pyloric orifice, which is the opening into the small intestine, and the pyloric sphincter, which is that muscular ring that's going to regulate the movement of food from the stomach to the duodenum into the small intestine. The histology of the stomach, you've got these simple columnar epithelia. They have these very deep gastric pits. So you can see this deep gastric pit all the way down in here. You've got gastric glands that are located in the base of the gastric pits, and the muscularis mucosa, very thin layer of muscle, is going to be able to contract, and that's going to help the gastric glands release their secretion. So what we're looking at here is a enlarged gastric pit. You've got 5 types of cells in the gastric epithelium. So you have 5 different types of cells. You can see those here. lining the gastric pits. 4 of those cells produce up to 3 liters per day of gastric juice associated with the stomach, and then the 5th type is a kind of like an endocrine cell that releases a hormone into the blood. So the surface mucus cells line the stomach lumen and they extend into the gastric pit. That's going to be the first group of cells that you see here. These are your surface mucus cells. They continually secrete an alkaline product. Alkaline is the acidic of the opposite of acidic, and it produces a mucin onto the gastric surface. So the mucin is again going to become hydrated with water and produce mucus. So basically what we're doing is producing an alkaline mucus layer. So if you remember earlier, I talked about how the stomach was well equipped to handle the acidity of its own environment, whereas the esophagus was not. And I said we were going to talk about how it's equipped to do that, and so this is where we're going with this. Those surface mucus cells release an alkaline mucus that helps to protect the remainder of the cells from the acidic environment of the stomach. The next cells are known as mucus neck cells, located just deep to the base of the gastric pit, and they're kind of interspersed among what are called the parietal cells. The mucus neck cells produce an acidic mucin, so it's a little bit different from the other mucin that was alkaline. The acidic mucin helps to maintain the acidic conditions resulting from the secretion of the hydrochloric acid that we're going to see from the parietal cells. The mucus produced from both the mucin that's getting released from the both types of the mucus cells has lubricating properties, so it helps to kind of lubricate that surface of the stomach lining. So here's our mucus neck cells, and then you kind of see they're kind of like interspersed later on as well. Then you've got parietal cells. These are your parietal cells. Parietal cells are responsible for releasing 2 important substances. One of those is intrinsic factor, which is required for the absorption of vitamin B12 in the ileum, which is the last portion of the small intestine. B12 is necessary for the production of normal erythrocytes. So if you get a decrease or an absence of B12, then you get something called pernicious anemia. The other secretion that's going to be released by the parietal cells is hydrochloric acid, or HCl. So that's going to be important because it's going to keep the environment of the stomach acidic. But it's important to note that it doesn't just make the HCl and then release it. Rather, it has to form the hydrogen and the chloride individually and then release those separately. If it created the hydrochloric acid within the cell and released it then, it would destroy the cell. It would be way too acidic. So it pumps out hydrogen and it pumps out chloride, and then once it's inside the gastric pit, then it combines to form hydrochloric acid, and we have acidic environment of the stomach. Also in the gastric pit you have chief cells. Chief cells release what we call zymogen granules. A zymogen is like an enzyme that's not yet active. It's kind of like an enzyme precursor. So specifically in this case we're talking about pepsinogen. Pepsinogen is the inactive precursor of the proteolytic enzyme pepsin. So what that means, let's kind of work backwards. Pepsin is an enzyme that degrades proteins. The word proteolytic means something that breaks down proteins. And so if we're talking about pepsinogen, it's just the precursor to pepsin. So we have to release them in these inactive forms for similar reasons to the reason why we had to do HCl the way we did. We can't just create these active protein degrading enzymes within the cell, because then it'll start just degrading all of the proteins in the cell and the cell would die. So we create it in this inactive form, pepsinogen, then it gets released into the gastric pit, and then it's actually going to be the hydrochloric acid that was released by the parietal cells that converts pepsinogen into pepsin. And so then it's activated. Now we've got an active enzyme that can start to denature some of the proteins in the stomach. Chief cells also produce gastric lipase. Okay, so here's that word again, lipase. So what are we breaking down? We're breaking down fat. Gastric lipase, because it's coming from the stomach, as opposed to the lingual lipase that we saw in the oral cavity. Here it's just got a very limited role in fat digestion. About 10 to 15% of the ingested fat will start to be released in the stomach by the gastric lipase. You'll see the majority of that's going to happen as we get to the small intestine. Some of the functions of hydrochloric acid that was again released by the parietal cell. It released hydrogen, it released chloride. Once it got into the gastric pit, those two combined to form the HCl. It converts the inactive enzyme pepsinogen into the active form, which is pepsin, and it provides an optimal pH environment for pepsin activity. So pepsin is an enzyme. It's one of the rare guys that's actually maximally functional at a low pH. So the more acidic the stomach, which it is because of the HCl, the more active the pepsin is going to be. It also kills microorganisms that enter the stomach. So most of the things that we ingest that would otherwise be harmful for us will die in the stomach because they can't survive that low pH. It contributes to the breakdown of plant cell walls and animal connective tissue, and then it denatures proteins by causing them to unfold, thus facilitating the chemical digestion by enzymes. So if you remember, proteins have that primary, secondary, tertiary structure. The tertiary is the three-dimensional folding of that protein. We can best digest them if they're unfolded, and then it's just a matter of chopping them down into amino acids. So hydrochloric acid helps to denature it, which means to unfold it, so we have a long chain of amino acids, and then our pepsin and other proteolytic enzymes can start chemically digesting them better. Lastly, we have the G cells. The G cells are what we call enteroendocrine cells. Entero because they're part of the digestive system, endocrine because they release a hormone. So you'll see those distributed throughout the gastric glands in the stomach. The G cells release something called gastrin. Gastrin goes into the blood and it stimulates stomach secretions and motility. So here's a summary of all of the cells and their products. Here's your surface mucus cells. This is going to be your alkaline mucus right here. Mucus neck cells are releasing a more acidic mucin here. Chief cells, you follow that up, it's releasing pepsinogen. Gastric, or your G cells, are releasing gastrin. That's going into the blood. That's going to increase release of secretory products and promote motility. Here's your parietal cell. It's releasing intrinsic factor. It's going to release chloride. It's going to release hydrogen so that those can go off and make hydrochloric acid. And then you can kind of see how everything interacts if you look at this top portion. So here's your protein in its three-dimensional structure. Here's the hydrochloric acid that's been released from the parietal cell. That's going to cause a denaturation of those proteins. So now they look more like this. They've lost their 3D structure. Then you had the pepsinogen, but remember the hydrochloric acid was responsible for converting pepsinogen to pepsin. So now you have this active proteolytic enzyme that is going to come act on these denatured proteins and break them down. And now you've got what we're calling oligopeptides. So see how this is a long chain of amino acids, and here we've got short chains. So it's just showing you the start of breaking down those proteins. Gastric mixing and emptying, so contractions of the muscle in the wall are going to mix the bolus of food with the gastric secretions to form chyme. So it's basically food when it goes in the mouth. We convert it to a bolus as it goes through the pharynx and the esophagus. When it gets to the stomach and it mixes with the secretions of the stomach, it's now called chyme. So you've got these peristaltic waves where it kind of starts up here and it moves down to the bottom of the stomach, and then that happens kind of over and over and over. It creates a pressure gradient where you get the buildup of the chyme down in the pylorus region. The pressure gradient forces that pyloric sphincter open, and you get a little bit of the movement of the chyme out of the stomach into the duodenum. So I'll say it now so I don't have to say it later. Duodenum is the first part of the small intestine. Some people call it duodenum. Some people call it duodenum. You can call it whatever you like. I call it duodenum. But I know a lot of times if you go to YouTube, you'll hear duodenum, so don't let it throw you off. It's the first part of the small intestine. Once some of the chyme comes into the small intestine, you get what's called this retropulsion, where the chyme kind of comes back and moves back superiorly for a second, and then you get peristaltic waves again coming down. So this allows it to move into the small intestine, bits and pieces at a time, but without maintaining the mixing. If we just moved it all down to the pylorus and we didn't have the retropulsion to move it back up, we wouldn't be continuously mixing the chyme with the secretions and enzymes. So now on to the lower GI tract. Everything before was the upper GI tract. It ends at the stomach, and then the small and the large intestine pick up after the stomach, and that makes up the lower GI tract. Also associated with the lower GI tract are your liver, gallbladder, and pancreas, which, as you'll see, all have a role in what's going to take place in the lower GI tract. So the process of digestion is going to continue in the lower GI tract, and more importantly, you're going to see that this is where the majority of the absorption is going to take place. So this is where we're actually going to complete the process of digestion and absorb these nutrients into our system, and then anything that can't be digested and absorbed is going to be eliminated at the terminal portion of the large intestine. So let's look at the small intestine. It's essentially a long tube. It's inferior to the stomach and located medially in the abdominal cavity. Ingested nutrients are going to spend about 12 hours in the small intestine. This is where we're going to finish the job of chemical digestion, and this is where the majority of the nutrients are going to end up being absorbed, and a large percentage of the water and electrolytes are going to get absorbed in the small intestine as well. Any vitamins that got ingested with the diet are also going to be absorbed as it goes through the small intestine. Gross anatomy of the small intestine: it's very coiled, thin-walled tube. It extends from the pylorus of the stomach all the way to the cecum of the large intestine. That's essentially the last part of the stomach to the first part of the large intestine. It occupies a significant portion of the abdominal cavity, and it's made up of 3 specific regions. The duodenum is the first segment of the small intestine. It originates at the pyloric sphincter, so as soon as that chyme exits the stomach, it's coming into the duodenum. It receives accessory gland secretions from the liver, the gallbladder, and the pancreas. The jejunum is the middle region of the small intestine. It's primary region where you're going to get chemical digestion, nutrient absorption, and then the final portion is the ileum. The distal portion terminates at what's called the ileocecal valve. So the cecum is actually the first part of the large intestine. The ileum is the last part of the small intestine. So in between those two, you have what's called the ileocecal valve. Sometimes you'll see it called ileocolic, but either one, it's the ileocecal valve. It's the valve that regulates the movement of the chyme as it moves out of the small intestine and into the large intestine. Absorption of digested materials continues through the ileum. So you've got your duodenum, this is your first part, and then it will become your jejunum, and then it becomes the ileum, and this around in here is where you would have your ileocecal valve as it comes into the cecum of the large intestine, which would be right about there. Okay, you have these circular folds of the small intestine. The mucosal and submucosal layers are kind of thrown into these circular folds, and that's why it has this kind of bumpy region. It serves a couple of different purposes. It increases the surface area. By having a high surface area, that increases the likelihood that we're going to absorb our nutrients. So we get maximal nutrient absorption because of these circular folds. They also act in a way like speed bumps to slow down the movement of the chyme. If everything were to rush through our small intestine really quickly, we wouldn't have enough time to complete the breakdown or the digestion and actually get absorption of the nutrients, and you would end up with malabsorption. So we need it to kind of take its time and move slowly through the small intestine so we can give those enzymes time to work, get everything broken down into the smallest component pieces, and then absorb that into our bloodstream. Histology of the small intestine, the epithelial layer itself has these finger-like projections. Those are called villi. You can see it in this slide. It kind of serves the same purpose as a circular fold. It increases the surface area that helps us to get maximum nutrient absorption. Each villus has a bunch of blood vessels that we need for nutrient absorption. So the blood vessels are going to run inside the villi so that as the nutrients come across, it can get into the bloodstream. You also have lacteals. So we did talk about lacteals in class. Those are those lymphatic structures that are necessary to absorb lipid molecules and lipid-soluble vitamins that would be otherwise too large to go into the capillaries. If you look at the actual cells themselves, they also have these extensions called microvilli. So it looks a lot like the villi. Same purpose, increase the surface area. So in the small intestine, we have maximally increased our surface area so that we can get maximum nutrient absorption. We've got glands in the small intestine that produce mucus, intestinal juices, and alkaline substances to protect from the acidic chyme. So just like our esophagus wasn't well equipped for the acidic environment of the stomach, neither is our small intestine. So we have to neutralize that chyme very quickly as it comes out of the stomach and into the small intestine. It's acidic as it's coming out, as it's coming out of the stomach into the small intestine, and we've got to neutralize it very quickly so that we don't damage the lining of the small intestine. And so we've got alkaline substances there to help with that. Motility in the small intestine is much like what we saw in the stomach. You've got the peristalsis, which is those contractions that are allowing you to move the bolus of food or the chyme from point A to point B. And then we've got segmentation. Segmentation is just like mixing that we saw in the stomach, but in the small intestine it's called segmentation. It's actually called something different in the large intestine as well, but it's all basically those kneading motions where we're kind of going back and forth, allowing the mixing of the chyme with whatever digestive enzymes are present at the time. So let's look at the liver. The liver is an accessory digestive organ. It's located in the right upper quadrant of the abdomen, just below the diaphragm. The main function, or the main digestive function of the liver is to produce bile. So it's the largest of our internal organs. It's made up of 4 partially separated lobes. You've got the right lobe, you've got the left lobe, you've got the caudate lobe, and you've got the quadrate lobe. The quadrate lobe is next to the gallbladder. So always remember the term GQ. You got G for gallbladder, Q for quadrate. So those two are next to each other. So then the next smaller one would be caudate. Notice in this picture that they have flipped the liver upside down. I don't really know how to say that. I don't want you to look at this picture and think that they turned it right and left. They flipped it up and down. So you can see how the gallbladder is on the bottom side here, and it's on the top side here. So when you're looking at the picture, orient yourself properly so you understand that they flipped it up. For the blood supply to the liver, you've got the hepatic artery. The hepatic artery is one of those branches that comes off the celiac trunk that we talked about in lab before. It carries oxygenated blood to the liver. The liver has connective tissue capsules that project inwards and divide the liver into different lobules, and each lobule serves as a functional unit, very similar to the bronchopulmonary segments that you see in the lung, but we've got lobules in the liver. On the periphery of each lobule, you have what's called a triad that's got an artery, a vein, and a bile duct. So you can see that here. You can see those 3 structures there. Within each lobule, you've got hepatic sinusoids that lie adjacent to the cords of hepatocytes, which again are just your liver cells, and then they've got macrophages in there to phagocytize anything that's coming in that we don't want, because remember, all of the blood from our GI tract goes straight to the liver, so we can kind of filter it to make sure there's anything we don't want before we send it to the inferior vena cava and goes back to the heart. So you've got these sinusoids, and then you've got macrophages there to take care of that. Then you've got the central vein that's going to drain the blood into the inferior vena cava. So this would be like a branch of the hepatic portal vein. This is blood that's coming in from the GI tract. It's going to go through these sinusoids. Anything we don't want is going to be phagocytized here, and then it's going to drain here and then go to the inferior vena cava, which is going to take it back to the heart. Cirrhosis of the liver is what happens when your normal hepatocytes end up being destroyed and they get replaced by this fibrous scar tissue. This happens whenever the liver is subject to chronic injury. It can be a product of alcoholism. There's certain liver diseases that lead to cirrhosis, and then certain drugs or toxins also as well. It can also be caused by chronic hepatitis, which is long-term inflammation of the liver. The early stages of liver cirrhosis are asymptomatic, so a lot of times you're not even going to know you have it until you've got this significant loss of function. Once the liver begins to decline, you're going to notice fatigue, weight loss, nausea. You may have some pain in the upper right quadrant. This is your normal liver, you can see, and this is your cirrhotic liver. The damage is irreversible. There's no way to kind of go back and undo it, which makes it unfortunate since a lot of times you don't even know you have it until you've kind of reached this later stage of cirrhosis. And the inferior portion of the liver is the gallbladder. The gallbladder is this sac-like structure that concentrates and stores the bile. So while the liver is the organ responsible for producing the bile, it will send it to the gallbladder for storage and concentration. And then as we eat a fatty meal, the gallbladder will contract and then release the bile into the small intestine. So bile, we'll talk about in a little bit, is something that we need for the digestion of fat. So you'll see a larger release of bile whenever you have a fattier meal. So gallstones can be formed when you get condensations of either cholesterol or calcium and bile salts. You can see the gallstones here in the picture. They can be anywhere from the size of a grain of salt all the way to the size of a golf ball. A lot of times you won't even know they're there unless they get lodged in the duct. So they could sit in the gallbladder, but as long as they're not in the duct and messing with the flow of the bile out of the gallbladder, then you may not even know they're there. But once they get lodged in that duct and it's hard to get the bile out, then you're going to notice them. Gallbladder pain is a lot of times hard to diagnose. It's pain perceived in the upper right quadrant, sometimes between the shoulder blades, sometimes causing nausea and vomiting. That's why, if you feel a pain in your shoulder blades, your first thought isn't always, it must be my gallbladder, or nausea and vomiting. That's not necessarily going to be your first thought. So sometimes it can take a while before somebody gets to, oh, it could be my gallbladder. The symptoms do typically get worse after eating a fatty meal because you're not able to easily digest it as much because you're not getting as much of the bile getting released. So treatment for this would be a cholecystectomy where you're going to remove the gallbladder. So I know if we were holding this in class, somebody would raise their hand and ask, so what happens after somebody has their gallbladder out? Can they not digest fat? The answer to that is you still can digest fat because remember the bile is being produced by the liver. You just don't have as much control over, okay, I just had this huge fatty meal, I can't get a huge amount of bile release because you don't have the storage container or the concentrator anymore because that would be the role of the gallbladder. So the advice is always to avoid those meals. When my husband had his out, his doctor said, eat what you want, and you'll figure out if it works or not. Because if you eat a meal and you don't have the bile to digest it properly, you're going to end up with some GI distress, and likely you won't be running to eat that meal next time. So yeah, you still can digest fat, it's just not as efficient. So you want to try to reduce the amount of fatty meals that you have because you're not going to digest them as efficiently. Another accessory structure that we have is the pancreas. The pancreas we've seen before because it has both endocrine and exocrine functions. So before we talked about the endocrine function, which was to regulate blood glucose level through insulin and glucagon. The exocrine function is to produce pancreatic juice, which assists with digestion. Remember the pancreas has the islet cells, which are the endocrine cells, and the acinar cells, which are the exocrine cells. The term pancreatic juice refers to a combination of secretions made by those acinar cells, and they go through the pancreatic duct. This is basically pancreatic juice, and you've got an enzyme to digest one of each of the following, biomolecules. You've got pancreatic amylase. So again, here's that amylase. We saw salivary amylase. That was amylase produced in by the salivary glands. This is amylase produced by the pancreas. So it digests starch. Pancreatic lipase. So far we've seen lingual lipase, gastric lipase, and now pancreatic lipase. So it digests the fats released by the pancreas. Inactive protease. So proteases digest proteins. They must be inactive because if they are active, as they get released through that pancreatic duct, it's just going to eat away at the cells of the duct. So these guys will become active once they make their way into the small intestine. And then nucleases digest nucleic acids, DNA and RNA, which is going to be in any plant or animal product that you eat. So if you look at the image, you can see here is your pancreatic duct, and you can see this is your common bile duct right here. Those two are going to merge and empty into the duodenum. So you're going to get pancreatic juice and bile all coming into the small intestine, and that's why we're going to get maximum digestion happening in the small intestine. Alright, regulation of accessory structures. Cholecystokinin is a hormone that gets released from the small intestine, mostly in response to fatty chyme. So if you eat a meal that's got more fat than usual, you're going to get the release of cholecystokinin. And the main job of cholecystokinin is to stimulate the smooth muscle in the gallbladder, so it's going to start to contract. So we're going to get the release of more concentrated bile. That bile is going to be important for breaking down that fatty meal. Also, we have secretin, which is released from the small intestine primarily in response to acidic chyme. So if we didn't neutralize it very well as it left the stomach and came into the small intestine and it's still very acidic, you'll get the release of secretin. Secretin causes the release of an alkaline solution that contains bicarbonate, or H2O3-, from the liver and the ducts of the pancreas, and then that's going to basically help to neutralize that chyme so we don't disrupt the lining of the small intestine. So finally, we're moving on into the large intestine. So this would be right here where I'm highlighting the ileum of the small intestine, and you can see it's going to come into the large intestine. This would be that ileocecal valve. The large intestine is a relatively short and wide tube. Because the majority of the nutrients got absorbed in the small intestine, the large intestine is mostly going to absorb water and electrolytes that are still remaining in the chyme. Whenever the chyme enters the large intestine, it's quite watery. The majority of that water is going to get absorbed by the large intestine. So as it's traveling through the large intestine and water is getting absorbed, that material is going to start becoming more solid until it becomes the fecal material that will be eliminated. The different regions of the large intestine, you've got three main regions. You've got the cecum, which is the first portion of the large intestine. It's located in the lower right quadrant. So here's the cecum here. It extends inferiorly from that ileocecal valve. And the chyme is going to come into the cecum from the ileum. Projecting inferiorly from the cecum is the vermiform appendix. So you can see that here kind of hanging off of the bottom of the cecum. The vermiform appendix is a thin, hollow, finger-like sac filled with lymphatic nodules. So it's a lymphatic structure. Okay, then you have the colon. The colon forms this inverted U shape and is divided into the ascending, transverse, descending, and sigmoidal colon. So please don't get confused. If somebody says, what are the regions of the large intestine? There's the cecum, the colon, and the rectum. If somebody says, what are the regions of the colon? You've got the A for ascending, transverse, descending, and sigmoidal. So kind of keep that in your mind. So ascending is where it goes up, and then you've got this flexure here. It's called the right colic flexure, and it takes a turn and becomes the transverse colon, kind of hooks upward a little and then flexes downward. Left colic flexure, this is called the descending colon. As it moves down, it starts to make this little curve structure right here. This is called the sigmoid colon there. And then the final portion is the rectum, muscular tube that expands to store the accumulated fecal material just prior to defecation. So you can see the rectum there. The anal canal is going to make up the last few centimeters of the large intestine. There you've got an internal involuntary sphincter and an external voluntary sphincter at the base to regulate the movement of the feces out of the body. Histology of the large intestine, you've got simple columnar epithelium. Unlike the small intestine, you don't have all the villi, you don't have all the extensive folding. That's because you don't have a ton of nutrient absorption that's going on in the large intestine. You're mostly absorbing water and electrolytes. You do have goblet cells in there that are secreting mucus, and then you've got lymphatic nodules as well. Bacterial action in the large intestine is called indigenous microbiota. This is the term for the normal bacterial flora that exists in the large intestine. They're pretty important. They break down the remaining carbohydrates, proteins, and lipids that might still be in the chyme. They produce carbon dioxide, hydrogen, and other substances, which is responsible for this foul smell. And then they produce B vitamins and vitamin K, which get absorbed from the large intestine into the blood. So that's one of their really important jobs, is to help with those two vitamins. Motility of the large intestine, you've got the peristalsis, where we're moving, the substance from point A to point B. It's a little bit slower, a little bit less powerful as it kind of flows through in the large intestine. Haustral churning, this is like the mixing motion or the segmentation. Mixing we saw in the stomach, segmentation in the small intestine, haustral churning for the large intestine, where we're just mixing what's there. And then you've got the mass movements, which are powerful peristaltic waves, and those are responsible for propelling the fecal material towards the rectum, so that's going to lead to an actual bowel movement. Colorectal cancer is the second most common cancer in the U.S., characterized by a malignant growth anywhere along the large intestine or the rectum. Usually they start from polyps, which are outgrowth from the colon mucosa. So a lot of people do have polyps that don't turn into cancer, but it's always important to monitor them because they always could. So you always want to monitor those polyps. Risk factors, family history, a low dietary fiber intake, and then age. The older you get, the higher your risk is. Your initial symptoms are going to present as bloody stool, change in bowel habits, which usually translates to constipation. Later, it can cause abdominal pain, fatigue, weight loss, and anemia. The prognosis depends on how early you catch it. If the cancer has only reached the mucosal layers, the prognosis is fairly good. If you don't catch it until it has reached the deeper layers or metastasis has already occurred, it's going to be a less optimistic prognosis. It's going to require a more aggressive form of treatment. And lastly, diarrhea and constipation. So, diarrhea happens when there's a disruption in the movement of water out of the large intestine. So remember how I said the chyme was very watery as it enters the large intestine? The way it's supposed to work is as we are moving that chyme throughout the large intestine, water is getting absorbed, and so we're forming this kind of semi-solid waste material. If it moves too quickly through there, or for some other reason we don't get the proper absorption of water, it stays very liquidy, and then you get diarrhea. This can also happen if you have osmotically active solutes in there, which are things that are going to draw fluid into them. So if everything was working the way it should, but then you introduce osmotically active substances and now water is leaving the blood and going into the large intestine, that would cause it to be watery as well. Constipation is a temporary inability to defecate due to compacted feces that are difficult to eliminate. So this could be caused by lack of fiber, dehydration, or anesthesia. Lack of fiber is going to mean that we don't have anything to draw fluid in. So the fluid has left, we're not getting any fluid back in, so it's harder to move along. Dehydration, you're just simply not hydrating enough to keep fluid in there to keep it moving. Anesthesia, because that's going to slow, that has more to do with the muscle. The muscles are going to move more slowly, so they're not moving it along as quickly. In any of those cases, it makes it more difficult to defecate.